So have you ever stopped to actually think about biology's ultimate problem? I'm talking about extinction, death. I mean, it is the absolute end of the line for literally every single living organism on this planet. And yet somehow, life has been thriving on Earth for billions of years. How is that even possible? How does life continuously manage to cheat death? Well, today we are going to unpack nature's most ingenious and, honestly, fundamental solution to this very problem.
Welcome to today's Explainer. I am absolutely thrilled to have you here because we're diving into one of the most fascinating mechanisms in all of biology. Yep, we're talking about reproduction. We're going to explore exactly how life continues, looking at everything from the microscopic wonders happening right in your kitchen all the way to massive, mind-blowing agricultural hacks used by farmers worldwide. All right, let's jump right in.
Okay, real quick, here is our roadmap for today. We'll kick things off with the cycle of life, look at the two paths of reproduction, and then dive headfirst into the sci-fi world of fission, budding, and fragments. After that, we'll check out the power of spores and wrap everything up with vegetative propagation.
Starting with the big picture, Section 1, the cycle of life.
To really appreciate reproduction, we have to grasp two very different but deeply connected concepts. First up is lifespan. This is just the absolute time period from the birth to the death of an organism. You're born, you grow, you mature, and eventually, well, those life processes stop. But to make sure a species doesn't just completely vanish when that lifespan ends, we have reproduction. This is the biological process where living organisms produce new individuals of their own kind. It's essentially the bridge between one lifespan and the next. Without it, the continuity of life on Earth just stops.
And bringing this down to earth, examples of this continuity are literally all around us. A parent mango tree gives rise to a seed, right? Which grows into a new tree. A cow has a calf. A dog has a puppy. A cat has a kitten. The overarching biological term we use for all these new individuals is offspring. Now, it's a simple, everyday reality, sure, but it perfectly highlights exactly why reproduction is necessary. It guarantees the continuity of a species, generation after generation.
But you know, nature isn't a one-trick pony. Let's move to Section 2, two paths of reproduction.
When it comes to making offspring, life essentially has two distinct strategies. First, we have asexual reproduction. This is nature's ultimate copy-paste function. It only requires a single parent. There are no gametes, so no sperm or ovum involved, and the offspring are genetically identical clones of the parent. Pretty wild, right? Then we have sexual reproduction. This one involves two parents and definitely relies on gametes. The offspring here inherit mixed characteristics from both parents, meaning they are totally unique, not identical clones. Today, we're really going to focus in on the fascinating, highly efficient world of asexual reproduction.
So, how does this actually look in the real world? Let's check out Section 3, fission, budding, and fragments.
Nature is absolutely packed with organisms that act as solo parents. We've got the amoeba, bacteria, yeast, and the hydra. But get this, consider the sponge. Now, you might look at a sea sponge and think, okay, that's a plant, or maybe that's a weird rock. But no, it's actually an animal. It's classified as a lower animal, but an animal nonetheless. And all of these organisms have completely mastered the art of cloning themselves. Let's see how they actually pull it off.
First up is binary fission. Imagine being able to just split yourself straight down the middle to create a whole new you. That's exactly what bacteria and amoebas do. It starts with the DNA replicating inside a single parent cell. Next, the cell elongates, and those genetic copies pull away to opposite ends. Then, a dividing wall, called a septum, forms right smack in the middle, and boom, the cell splits into two equal, identical daughter cells. Literally, one parent becomes two offspring in a matter of minutes or hours.
But sometimes, splitting into just two isn't enough, especially when the environment gets tough. Enter multiple fission. Instead of dividing into two, one single cell divides into many daughter cells simultaneously. The amoeba does this when it faces unfavorable conditions, kind of like an emergency survival tactic to maximize the chances of its clones making it. The malarial parasite Plasmodium also uses this exact rapid-fire cloning technique.
Let's pivot to a bit of kitchen science. Picture this. You take a test tube with a bit of warm sugar solution, drop in a pinch of yeast granules, plug it up, and wait an hour. What exactly is happening in there under the microscope?
Well, if you looked closely at that yeast, you'd see our next method in action: budding. Budding is when a small outgrowth, literally a bud, forms right on the parent's body. The parent feeds it, it grows, and once it's mature enough, it just pops off to live its own life. Yeast does this super rapidly in sugar water, and that animal we mentioned, the hydra, does it in ponds. Then we have fragmentation. If you've ever noticed that green, stringy algae in a pond, like Spirogyra, that organism reproduces by literally breaking its own body into pieces. Each severed fragment then regenerates and grows into an entirely new, fully functioning individual. Crazy, right?
All right, moving on to Section 4, the power of spores. This is a microscopic wonder that might actually be happening in your pantry right now.
So here is a classic observation you can do at home. Moisten a slice of bread, toss it in a dark, warm box, and just leave it. Within days, you're going to see mold growing. But where did it come from? It didn't just magically appear. See, invisible, incredibly lightweight airborne spores are constantly floating around us. When they happen to land on that moist, warm bread, they hit the jackpot. They sprout this network of threads to absorb nutrients, and eventually they form tiny sacs that burst, releasing countless new spores right back into the air to start the whole cycle over again.
Let's quickly define the anatomy of this bread mold, which is technically called rhizopus. First, you have the sporangium. This is that round, sac-like structure at the very tip of the mold. Think of it like a little container packed full of those tiny reproductive spores. Down below that, actually anchored into the bread, is the mycelium. This is a vast, spreading network of thread-like structures or hyphae that reaches over the food to steal all its nutrients.
Now, for a really long time, people honestly thought mold just spontaneously generated out of thin air. But the brilliant scientist Louis Pasteur proved otherwise. He famously demonstrated that life comes only from pre-existing life, not from non-living matter. He used this special swan neck flask filled with nutrient broth. As long as the flask was sealed off from dust, absolutely nothing grew. But the very moment he broke the neck and let the outside air in, the broth filled with life. He essentially proved that the spores were floating in the air all along.
At this point, you might be thinking, okay, great, fungi ruins my bread, who cares? But fungi are actually incredibly important. First off, they are nature's ultimate recyclers, breaking down organic waste. But it gets even better. That mycelium network we talked about, it can actually absorb and remove toxic heavy metals from industrial waste, effectively cleaning our environment. And perhaps most importantly, fungi are absolutely crucial for modern medicine. Life-saving antibiotics, like penicillin, are derived directly from fungi to fight off bacterial infections.
Okay, for our final section, we are stepping away from fungi and microbes and looking at the plant world. Section 5. Vegetative propagation.
It is honestly incredible to see how different parts of a plant can naturally spawn entirely new life without ever needing a seed. Take the bryophyllum plant, for example. Its leaves literally grow tiny new plants right along their edges. And here's a fun fact. It's sometimes called the stonebreaker plant because it's actually used in Ayurvedic medicine to help treat kidney stones. Then you have underground stems, like potatoes and ginger, which sprout new shoots right from their eyes. And even roots, like the sweet potato, can develop buds that grow into fresh plants. Nature just does this completely on its own.
But humans, specifically farmers and botanists, have figured out how to hack this system through artificial vegetative propagation. And there are 3 main techniques we use. Cutting is super simple. You just take a piece of a stem and plant it in soil to grow a new plant. Layering involves bending a live branch down to the ground, burying part of it in the dirt until it grows its own roots, and then cutting it free. And then there's grafting, which is this incredibly clever technique of physically joining the tissues of 2 completely different plants together so they heal and actually grow as one single plant.
Let's look a little closer at how grafting actually works, because it's fascinating. Imagine you have a plant that produces these beautiful pink flowers, but has a really weak root system. And then you have another plant with incredibly strong, hardy roots, but yellow flowers. You can actually combine them. You take the top stem of the pink flower plant, which we call the scion, and the rooted base of the yellow flower plant, the stalk. You make diagonal cuts on both, bind them tightly together with tape, and just wait. The vascular tissues eventually heal and merge together. So you end up with a single superplant utilizing the absolute best traits of both, all through asexual cloning.
So, we've covered a lot of ground today, right? From the splitting of microscopic amoebas to the grafting of massive fruit trees. But I want to leave you with a final thought to really ponder. If an amoeba splits into 2 identical pieces, and those pieces split into more identical pieces, and the DNA remains exactly the same generation after generation, did the original parent ever really die? I mean, if asexual organisms are perfect, unbroken, identical clones of their ancient ancestors, are they technically immortal? Think about that for a second. Thank you so much for joining me in this explainer. Keep learning.